Hello students, today in this presentation you people are going to learn the topics DNA as genetic material, DNA structure and DNA replication. First, I will start explaining the topic DNA as genetic material. To get it studied, it is better to introduce the knowledge of how and why a nucleic acid like DNA was considered as the genetic material. This is followed by the experiments of Griffith, Avery team, Hershey and Chase experiments. So first let us come to know the introduction part. In the first half of the 20th century, Gregor Mendel's principles of inheritance became widely accepted. But the chemical nature of hereditary material remained unknown. Based on his experimental investigations, Mendel concluded that development of characters were due to the presence of particular factors which were called as genes, and these particular factors were transferred from one generation to another generation. However, the chemical nature of genes fascinated the scientists for many years. But during 1920s, a series of experiments conducted by scientists finally revealed that DNA was the genetic material. But here we have a question: what decide which molecule can be a genetic material? There were certain properties. Whichever molecule satisfy these properties can be a genetic material. So let us come to see the properties of genetic material. These are the properties of genetic material. The hereditary information must be there in the structure of the genetic material. It should undergo replication. It should be able to express itself through the formation of specific biomolecules, and it should be able to undergo mutations, and such a change should be stably inherited to the next generations. Having seen the properties of genetic material, we will now focus on Griffith's experiment. Regarding this, it was in 1928. Frederick Griffith conducted a basic experiment on bacterial transformation. In the process of identification of DNA as the genetic material, Griffith conducted experiments on Diplococcus pneumoniae, a bacterium which causes pneumonia, and this is also known as Streptococcus pneumonia. In his experiments, Griffith used two strains of Streptococcus pneumonia. One is called S3 strain and the second one is R2 strain. Coming to know the properties of S3 strain. These bacterial cells have smooth polysaccharide capsule and they are capable of producing smooth colonies. It is a virulent bacterium. That means it is able to cause pneumonia. Because of the presence of this smooth polysaccharide capsule, it is protected in the host immune system and able to cause pneumonia. When it comes to see R2 strain properties, these bacterial cells do not have smooth polysaccharide capsule. They are capable of producing rough colonies. It is a non-virulent strain. Non-virulent means non-pathogenic strain. They are unable to cause pneumonia. Because of the absence of this smooth polysaccharide capsule coat, they will be damaged in the host immune system and unable to cause pneumonia. During the course of his experiments, Griffith injected mice with S3 strain. Mice developed pneumonia and died. Then, when mice were injected with the R2 strain, there were no signs of pneumonia because it is a non-virulent strain. Then, he killed S3 strain by subjecting it to heat treatment and injected into mice. there were no symptoms of pneumonia, indicating that all the cells were killed by heat treatment. Then he tried a combination of heat killed S3 strain and mixed them with live R2 cells and injected into mice. Surprisingly, there were symptoms of pneumonia. Here we get a question: if it is a heat killed bacterium, will it be able to cause disease? The answer to this question was like this: analysis of dead mouse tissue was found to contain live R2 cells which had smooth polysaccharide capsule like S3 cells. This is the diagrammatic representation of Griffith's experiment. And what did this experiment prove? And what must have gone into the R2 cells? From the results, Griffith concluded that there was something that was transferred from heat killed bacterium into R2 cells which helped them to synthesize a smooth polysaccharide capsule and became virulent. This was called as bacterial transformation by Griffith. That means one bacterium was changed into another bacterium. So here non-virulent bacterium was transformed into virulent and the substance that was transferred from virulent bacterium into non-virulent bacterium was known as transforming principle by Griffith. So today we know transformation is a specific type of recombination. A segment of DNA replaces another homologous segment on bacterial chromosome. However, Griffith's experiment was a turning point towards the discovery of DNA as the genetic material, but he could not identify the biochemical nature of transforming principle in his experiments. Now, the focus was on finding out the biochemical nature of transforming principle in Griffith's experiments. Many scientists performed different experiments. There were different observations, results and opinions. A team of scientists, Avery, McCall and McCarthy, performed experiments to determine the biochemical nature of transforming principle in Griffith's experiments. So for this, they first extracted various biochemicals like carbohydrates, proteins, lipids, RNA and DNA from heat killed S3 strain. Then these extracts were subjected to treatment with different enzymes and alcohol before they were mixed with live R2 cells and injected into mice. The observations and results were recorded like this. First, the cellular extracts were mixed with alcohol. They observed that it got precipitated by alcohol. In the second part of the experiment, the cellular extracts of S3 strain were subjected to treatment with protease enzymes, lipases and RNases and injected into mice. It could cause transformation. That means mice developed pneumonia and died. Then the scientist subjected the cellular extracts of S3 strain with the DNase enzyme and mixed this preparation with live R2 cells and injected into mice. It could not cause transformation, indicating that the transforming ability was lost. Then what could be the transforming principle? It was confirmed by the researchers it was not a carbohydrate, not a protein, not a lipid, not RNA. How could they confirm this? being a carbohydrate, it would have not got precipitated by alcohol. So, it is very clear it was not a carbohydrate. Proteases are enzymes with which proteins will get damaged. Lipases are enzymes which destroy lipids. RNases are enzymes which destroy RNA. But even in the absence of these biomolecules, transformation happen. So, it is very clear that these biomolecules were not responsible for transformation. Then the scientists were left with one option to decide which biomolecule is responsible for transformation. It was DNA. After treatment with the DNase enzyme, transformation could not happen. There were no signs of pneumonia in mice, indicating that the transforming principle was lost. Then what did this experiment provide? What did this experiment show? The genetic material that is getting passed on from one bacterial cell to another bacterial cell was nothing but DNA. So, DNA can transform the properties of a cell. Here in this case, it was the appearance and pathogenicity of a bacterium, and the results of these scientists were published in 1944. But this experiment was not accepted by most of the biologists due to the lack of information on the molecular mechanism of transformation. So, finally, a confirmatory report came from the experiments of the scientists Hershey and Chase in 1952. Hershey and Chase, this is also known as Hershey and Chase blender experiment. They took T2 bacteriophage, which is a virus that infects the bacterium E. coli, Escherichia coli. You have to remember one point, T2 phase itself has only two things in its structure, DNA and protein coat. During the infection of bacterial cells, T2 phase injects only its DNA into the bacterial cell, but the protein coat always remains outside the host cell. And inside the bacterial cell, the DNA molecule undergoes replication and forms new viruses. This is the life cycle of T2 phase. So, remember this. In their experiments, Hershey and Chase had grown T2 phases in two different mediums containing two different types of radioisotopes. In the first sample, T2 phages were grown in medium containing sulfur 35 radioisotope to label the proteins of viruses. In the second sample, the bacterial cells, the viruses were grown in medium containing phosphorus 32 radioisotope to label the DNA. This is the experimental setup of Hershey and Chase. So, here it clearly shows that in the first part of the experiment, T2 phages were grown in medium containing sulfur 35 radioisotope to label the protein coats of the viruses. This happens because sulfur is a component of certain amino acids. So, it is incorporated into the proteins during protein synthesis. When these sulfur 35 labeled protein viruses were used to infect these E. coli bacterial cells, the protein coats were left outside the host cell. That means, radioactivity was left outside the host bacterial cells. Then the non-labeled DNA undergoes replication and forms new viruses. Now, coming to see what is happening in the second part of the experiment. In the second part of the experiment, T2 phages were grown in medium containing phosphorus 32 radiolabeled viruses. Because phosphorus is a component of DNA, so it is incorporated during DNA synthesis. So, the nucleic acids DNA is labeled with phosphorus 32 radioisotope. When these P32 labeled viruses were used to infect E. coli bacterial cells, radioactivity in the form of DNA enters the host bacterial cells, but the non-labeled protein codes were left outside the host cell. Now, within the host cell, these radio-labeled DNA undergoes replication and forms new viruses. After infection, viruses and bacterial cells were separated by blending and centrifugation and then observed for radioactivity by the scientists Hershey and Chase. Now, have a look at this slide. Bacterial cells, you can see bacterial cells were formed as a pellet at the bottom of the test tube when treated with phosphorus 32 radio-labeled viruses. That means radioactivity was inside the bacterial cells. But in case of viruses that were treated with sulfur 35 radio-labeled viruses, radioactivity was outside the host cell. That means smaller virus, after centrifugation, smaller viruses remained in the supernatant, but the bacterial cells formed in the bottom of the centrifuge tubes. Now, I am talking about the radioactivity. The radioactivity was outside the bacterial cells when treated with sulfur 35 radio-labeled viruses, but it was inside the bacterial cells when treated with phosphorus 32 radio-labeled viruses. So, coming to know the conclusion, results of this experiment. What did this experiment provide? This experiment provided a strong evidence that DNA was the genetic material in T2 phases. How can the scientist explain this? How could they explain this? It can be explained like this. During infection of E. coli bacterial cells, all the protein codes were left outside the host bacterial cells. They didn't enter the bacterial cells. So, that means they were not responsible for the formation of new viruses, but it was only DNA that entered the host bacterial cells, and as it is having the genetic information of T2 phases, it was transferred from one phase generation to another phase generation. So, this way the findings of Hershey and Chase led to the universal acceptance of DNA as the genetic material. Next, I am going to talk about DNA structure. The things you will study here are introduction, key components present in DNA structure, Watson-Crick double helical structure of DNA. Let us come to see the introduction part about the discoveries of DNA. We have seen how DNA acts as the genetic material, but still there are many questions to be answered, like how DNA stored the genetic information and how is DNA copied so easily. These questions would be answered by discovering the structure of DNA. DNA or deoxyribonucleic acid was first identified by a Swiss chemist Frederick Mischer in 1869 in the nuclei of human white blood cells. It was about in 1953 James Watson, Francis Crick, Maurice Wilkin, Rosalind Franklin, using a powerful technique called X-ray crystallography, figured out DNA as a double helical structure. It is a twisted ladder, and they revealed that this structure could carry the biological information. In 1962, Watson Crick, Maurice Wilkin received a Nobel Prize for their discoveries concerning to the molecular structure of DNA, but unfortunately Franklin died of cancer 4 years before these researchers received the Nobel Prize. So, here at this point we will discuss the structure of DNA. DNA is composed of 2 polynucleotide chains. Each polynucleotide chain is made up of monomeric subunits called nucleotides. Now, let us have a look at the structure of nucleotide. Nucleotides are the building blocks of DNA. Each nucleotide is made up of 3 components: a 5-carbon sugar, a pentose sugar, 1 or more phosphate groups, and a nitrogenous base. Now, we will move to discuss the properties of pentose sugar. It is a deoxyribose sugar. The oxygen atom present at carbon atom 2 of ribose sugar is missing. So, it gets the name deoxyribose sugar, and DNA has the name deoxyribonucleic acid, and it is attached to the 3' carbon and 5' carbon of this pentose sugar are attached to a phosphate molecule to form a phosphodiester bond, and the 1' carbon is attached to a nitrogenous base by means of a beta N glycosidic bond. It is an inorganic phosphate in DNA structure. It has the formula H3PO4, and it contains 3 reactive groups, 3 reactive hydroxyl groups. 2 of them are involved in the formation of sugar phosphate backbone in DNA. Why we know DNA as an acid? Acidic nature of DNA is due to the presence of this phosphate group. Here we have the structures of deoxyribose sugar and phosphate group. You can see oxygen atom present at carbon atom 2 of this deoxyribose sugar is missing. This property is important for the enzymes to recognize DNA and RNA in the living cells. This is inorganic phosphate having 3 reactive hydroxyl groups. 2 of them are involved in the formation of sugar phosphate backbone in DNA, that we will discuss in the next slides. Next, coming to know the nitrogenous bases in DNA structure. Different types of heterocyclic nitrogen ring containing compounds are present in DNA. These are of 2 types, pyrimidines and purines. Now, we will discuss about pyrimidines. It is a single ring containing compound, and purines are of 2 types in DNA, thymine and cytosine. Thymine is a 2,4-dioxy-5-methyl pyrimidine, whereas cytosine is a 2-oxy-4-amino pyrimidine. Here we have the structures of cytosine and thymine. They are read in clockwise directions, and they are similar to benzene ring, except that it is having nitrogen at positions 1 and 3. Coming to know the structures of purines. They are double ring compounds containing a 6-membered pyrimidine ring and a 5-membered imidazole ring. Purines, they are of 2 types in DNA, adenine and guanine. adenine is a 6 amino purine, whereas guanine is a 2 amino 6 oxy purine. Now have a look at the structures of adenine and guanine. Both are double ring containing compounds, and you can see that they are fused at positions 4 and 5 carbon atoms, and they are having nitrogens at positions 7 and 9. They are read in anti-clockwise direction. Once we know the structural components in DNA, we must have to focus on the differences between nucleosides and nucleotides in DNA. Nucleoside is formed when a deoxyribose sugar joins to a nitrogenous base, whereas a nucleotide is formed when a nucleoside joins to one or more phosphate groups. So, obviously, there are 4 types of deoxyribonucleosides in DNA. These are deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine, and the nucleotides in DNA are deoxyadenilic acid, deoxyguanilic acid, deoxythymidilic acid, and deoxycytidilic acid. Naming of nucleotides is based on the presence of number of phosphate groups. Here this is a nucleotide structure and it is having only one phosphate group, so it is named as deoxynucleoside 5' monophosphate DNMP. If it contains 2 phosphates, it will be named as diphosphate. If it is having 3 phosphates, it will be described as triphosphate. A nucleotide joins to another nucleotide by means of a phosphodiester bond. A phosphate molecule links the 3' carbon and 5' carbon of adjacent sugar rings like this. This is one type of condensation reaction, and two hydroxyl groups of phosphate molecule are involved in this bonding. So, in this way this results in the formation of a dinucleotide. So, like this number of nucleotides are joined together to form a polynucleotide chain. So, this polymer is what we know as DNA. Now, coming to know Watson-Crick double helical structure. Watson-Crick suggested that DNA is composed of 2 polynucleotide chains, 2 strands of nucleotide chains that twisted around each other to form a double helix. It is a right-handed helix. That means if you hold it like this, it will they will twist around each other to form a double helix in clockwise direction. It is referred as B form. The 2 strands of DNA molecule run in opposite direction. That means they are anti-parallel. One strand runs in 5' to 3' direction, the second strand in 3' to 5' direction, and the 2 strands are held together by hydrogen bonds that are formed by complementary base pairs. The base pairing in DNA is very specific. If one strand has a purine base, the opposite strand will have pyrimidine base. This follows Chargaff's rule, and the bases are joined by hydrogen bonds like this. Adenine pairs with thymine by means of 2 hydrogen bonds. Guanine pairs with cytosine by means of 3 hydrogen bonds. How exactly does this take place? This is due to the spatial configuration and arrangement of atoms in each base, so that the bases will pair only in certain combinations, and this is helpful to maintain a fixed distance between the 2 strands, and this allows something called complementary base pairing in DNA. That means both strands are not identical. This is Watson-Crick double helical structure of DNA. It is looking like a spiral staircase with complementary base pairing, major, minor grooves, and showing diameter and other properties that we will discuss in the next slides. This structure is showing how sugar phosphate backbone is projecting outside of the DNA molecule and complementary base pairings are stocked inside the DNA molecule. Because of the presence of these large number of hydrogen bonds, there is a high degree of stability in DNA molecule, and these properties are important concerning the mechanism of replication in DNA. And the order of these sequences, base sequences, determine genetic code in DNA. These are other properties of the double helix. Diameter is 20 angstrom equal to 2 nanometer. It has a pitch, that means length of that helix to complete one turn is 34 angstrom, that means 3.4 nanometer, and there are 10 base pair per each turn of the helix. The distance between each base pair is 3.4 angstrom equal to 0.34 nanometer, and the helix has major and minor grooves which are formed as a consequence of arrangement of bases. What is the importance of DNA structure? From this, it is clearly understood that DNA stores the genetic information, it undergoes replication, transfer the information to the next generation, and express through the formation of specific biomolecules only because of its unique structure. Next, I will be talking about DNA replication. Here, you will come to know the definition of DNA replication, key enzymes involved in replication, and the mechanism of DNA replication. What is replication? Replication is the process of making copies of DNA. The purpose of replication is to produce two identical copies of DNA. And we know DNA regulates the activities of a cell because of its unique structure. So, when the cell divides, DNA should also duplicate to be able to get distributed equally to the daughter cells. That means, the genetic material must undergo replication. What are the types of replication? Three types of replication have been proposed at the molecular level: conservative, semi-conservative and dispersive. But it was experimentally proved by Messelson and Stahl experiment, DNA replication was semi-conservative. But we are not concentrating on this experiment, we are mainly focusing on the mechanism of DNA replication. If replication is conservative, one daughter molecule will have 2 whole strands and the second daughter molecule will have 2 newly synthesized strands. If replication is semi-conservative, each daughter molecule will have 1 parental strand, that means 1 whole strand and 1 newly synthesized strand. If replication is dispersive, each daughter molecule will have combinations of parental strands and newly synthesized strands, but DNA replication is semi-conservative. Next, we will have to focus on the key enzymes involved in DNA replication one by one. First one is helicase enzyme. Before replication, DNA needs to be unwound. That means both strands of DNA should be separated from one another. So, this allows DNA polymerase enzyme to find its substrate and replication can take place. This is done by the enzyme helicase, which breaks the hydrogen bonds and separates the 2 strands of DNA. And next one is gyrase enzyme. This enzyme prevents the DNA ahead of the replication fork and removes the tension that is created during unwinding process. And next one is single stranded stabilizing proteins. What these proteins will do is they bind to the single strands thus produced and stabilize them, that means hold them in place. Next one is primase enzyme. This enzyme catalyzes the synthesis of a primer. This is very important in DNA replication. Primer serves as a starting point in DNA replication because the enzyme DNA polymerase which catalyze the synthesis of new DNA strand always requires free 3' hydroxyl group to add new deoxyribonucleotides. So, this is provided by the strand RNA strand primer. Primer is a RNA strand synthesized by the enzyme primase. Now, DNA polymerase it catalyzes new DNA strand synthesis. Ligase enzyme joins the DNA strands and fills the gaps. Coming to know the details of DNA polymerase enzyme. This enzyme was discovered by the scientist Arthur Kornberg in 1956, and it directs DNA synthesis in 5' to 3' direction, but it always requires free 3' hydroxyl groups. It adds new nucleotides only to a pre-existing strand. We are now going to talk about 3 types of DNA polymerase enzymes, polymerase 1, 2 and 3. Coming to know their properties. 5' to 3' polymerization activity is very important in the extension of primer, that means adding new nucleotides to the primer, and 5' to 3' exonuclease activity it removes primer in this 5' to 3' direction. 3' to 5' exonuclease activity provides proofreading function, that means if wrong nucleotides are added to the new strand, they will be detected and removed by this activity of DNA polymerase 1. And next one is DNA polymerase 2 and DNA polymerase 3. Both these enzymes are having 5' to 3' polymerization activity and 3' to 5' exonuclease activity. 5' to 3' exonuclease activity is absent in these enzymes. It is present only in DNA polymerase 1 enzyme, and this is important in the removal of primer. And DNA polymerase 3 is the main enzyme of DNA replication. Now, let us come to know the steps involved in DNA replication. DNA replication involves 3 steps: initiation, elongation and termination. Coming to know the initiation process. DNA replication always begins at a unique point called origin of replication site, ORIC, and it is a sequence of about 300 base pairs in most of the prokaryotes with highly conserved sequences, and it consists of 3 repeats of 13 base pair sequences and 4 or 5 repeats of 4 base pair sequence sites. They will be recognized by a specific initiation protein complex. First of all, DNA protein binds to this repeat. So, this binding facilitates unwinding of DNA, and next DNA B protein binds to this repeat, and this facilitates unwinding of DNA because this is helicase enzyme. It breaks the hydrogen bonds and opens the 2 strands so that replication can take place. Next, this is facilitated by DNA C protein. Unwinding of DNA strands results in the formation of 2 Y-shaped replication forks in case of bidirectional replication, and these forks move in opposite direction as the replication process proceeds. They move down the DNA, and finally an enzyme primase DNA G catalyzes the synthesis of primer. Primer is a RNA strand. This is the diagrammatic representation showing the formation of initiation complex by DNA A and DNA B proteins, and This is unwinding of DNA double helix by helicase enzyme. Gyrase enzyme removes the tension stress that is created during unwinding process. Single stranded stabilizing proteins, by binding to the single strand, stabilize them so that each strand can act as a template for the formation of a new complementary DNA strand. Primase enzyme catalyzes the synthesis of a primer to provide free 3' hydroxyl groups. Next step of DNA replication is elongation. Once the primer has been synthesized by primase enzyme, next DNA polymerase 3 takes the place and adds new deoxyribose nucleotides to the free 3' hydroxyl groups of the primer. Here we must know what is meant by leading strand. In case of replication, that is taking place on the template strand which is in the 3' to 5' direction, new strand is synthesized continuously in the 5' to 3' direction. So, here a single primer is required. In case of replication, that is taking place on the template strand which is in the 5' to 3' direction, replication of the second strand is discontinuous. As it is discontinuous, it is called lagging strand. So, multiple primers are here required to start each fragment. These discontinuous fragments that are made are called as Okazaki fragments in the name of the discoverer Rizzi Okazaki. So, later these short fragments are joined together by the enzyme DNA ligase. That means one strand is synthesized continuously, new strand is synthesized continuously in the same direction of the replication fork movement, that is called leading strand. Whereas the second strand called as lagging strand is synthesized discontinuously in the form of short fragments called Okazaki fragments in the opposite direction of the replication fork movement. By looking at this diagram, you will clearly see how replication is taking place on both the strands simultaneously. One strand is called leading strand because it is synthesized continuously. The second strand is called lagging strand because it is synthesized discontinuously. It is in the opposite direction of the replication fork moment. So, multiple primers are synthesized at specific sites because of the non-availability of free 3' hydroxyl groups at the direction of the replication fork moment, and replication is taking place by the enzyme DNA polymerase 3. Next, coming to know the removal of primer by the enzyme DNA polymerase 1 using its 5' to 3' exonuclease activity. After synthesizing the primer by primase enzyme, DNA polymerase 3 adds nucleotides until it reaches the previously Okazaki fragment. There it stops and dissociates. Then this place will be taken by the enzyme DNA polymerase 1. DNA polymerase 1 using its 5' to 3' exonuclease activity removes the primer. Here we get a question, why primer is required for DNA synthesis and later it is removed? Because primer provides free 3' hydroxyl group without which DNA polymerase enzyme cannot initiate new DNA synthesis. But being a RNA strand, it should not be there in the DNA molecule. So, it is removed by the DNA polymerase 1 with its 5' to 3' exonuclease activity, and the gaps are sealed by gaps are actually replaced by new DNA strand by the polymerization activity of this enzyme, and then their gaps are sealed by the enzyme DNA ligase. And meanwhile, proofreading is done by 3' to 5' exonuclease activity of this enzyme DNA polymerase 1. So, this slide is showing the mode of action of DNA polymerase 1, how it is removing primer RNA nucleotides, and the gaps are sealed by the enzyme DNA ligase. So, DNA ligase now seals the gaps by the formation of a phosphodiester bond to form a continuous strand. DNA ligase enzyme ligates the 5' phosphate and 3' hydroxyl groups. This is the end of DNA replication. The replication forks will have to terminate and stop DNA synthesis at a specific point. So, this is done at a particular sequence called termination sequence sites. This is just at the opposite side of the origin of replication, and this termination sequence will be identified, recognized by a protein called TUS protein, terminus utilization substance protein. TUS and TER act as a barrier that prevents leading strand and that pauses leading strand until lagging strand arrives from opposite direction. So, this is done when helicase touches the TUS protein. So, this induces termination of DNA. Now, the DNA polymerase III enzyme synthesizes complementary strands on both sides of the replication fork. This is end of DNA replication. So, termination is taking place at TUS and TER sequence. This is the scheme of DNA replication. So, it is showing the moment of replication fork and how primers are synthesized and how single stranded stabilizing proteins stabilize the strands. Gyrase enzyme removes topological tension. This is because this is a specific example of the enzyme topoisomerase, and helicase enzyme unwinds the DNA by breaking the hydrogen bonds, and one strand is synthesized continuously. Here it is requiring a single primer, and another strand is synthesized discontinuously. This is called lagging strand. Here you can see multiple primers are synthesized, and then they will be sealed by the enzyme DNA ligase. Actually, synthesis of lagging strand is a bit later than that of leading strand. So, to get coordination between these two strands, the lagging strand will be pulled out of between the replication fork and DNA polymerase III enzyme. Then coming to know what is the result of DNA replication. Replication results in the formation of two identical DNA molecule. Each molecule will have one parental strand and one newly synthesized strand. So, this is actually proving that DNA replication is taking place by semi-conservative mode. So, this is all about DNA replication.